My name is Hilmar, and I'm the senior author of a study that is published in Science on October 9th. The title of the paper is Coupling of NMDA receptors and TRPM4 Guides Discovery of Unconventional Neuroprotectants. In this video I will tell you in less than 4 minutes why this study may be of interest to the general public. In a nutshell, this study is about neurodegeneration and the discovery of a new class of molecules, small molecules, no bigger than aspirin or ibuprofen, that can effectively prevent neurodegenerations. Now, why is that important? It's important because neurodegenerative diseases are very common disorders. They represent a major health problem, especially in the aging Western society. They're causing human suffering as well as substantial socioeconomic costs. And here's what makes them particularly devastating. There is no treatment, neither for Alzheimer's disease, Parkinson's disease, Huntington's disease, or amyotrophic lateral sclerosis, better known as ALS, nor for stroke or traumatic brain injury. Our study could possibly change this. Not necessarily today or tomorrow, but having uncovered the molecular basis of a cell death process that is common to virtually all neurodegenerative conditions, there's hope that we can now develop effective treatments to fight those diseases. And having already identified small molecule neuroprotective drugs, this could speed up this process enormously. Now, very briefly, what is the basis for this new therapeutic principle? It is very simple, and the story goes like this. It is all about a particular neurotransmitter receptor, which is called the NMDA receptor. This is arguably one of the most important receptors in the mammalian brain because it controls virtually all adaptive processes in the nervous system. And this includes in particular our ability to store information and form memories. But this is only true for NMDA receptors that are localized to the synapse, which is the site of communication between nerve cells. But NMDA receptors are not only present in synapses, they're also found outside synapses on so-called extrasynaptic locations. And this is where NMDA receptors turn evil, switch from Dr. Jekyll to Mr. Hyde. So if extrasynaptic NMDA receptors are activated by the neurotransmitter glutamate, they kill neurons. And this is what happens in many neurodegenerative diseases. But why do they kill neurons? Now they do so, and that's the key finding of our study, because at extrasynaptic locations, NMDA receptors form a death complex with another protein, which has the name TRPM4, which is pronounced TRPM4. The new class of molecules that we found act like a wedge that tear apart the complex, thereby stripping off the toxic activity from extrasynaptic NMDA receptors. But do these molecular wedges, which we named interface inhibitors, really help in neurodegenerative diseases? Yes, they do, although so far we have tested them only in mouse models of stroke and retinal degeneration. But it is likely that the same therapeutic principle may also be effective in humans. Now this could be a game changer, allowing for the first time to mitigate currently untreatable human neurodegenerative diseases.